Welcome and thank you for joining today's webinar, Mitigating Target Interference Challenges in Bridging Immunogenicity Assays to Detect Anti-Tocilizumab Antibodies. I'm Jessie Ricks, and it's a pleasure to introduce this important topic. To provide some background, Somru Bioscience is an emerging biotechnology company focused on developing breakthrough antibody technology research, diagnostics, and therapeutic applications. Our proprietary Somru technology enables production of antibodies with superior binding affinity and bioactivity across various biological assays in a short period of time. Our value proposition is built on extensive scientific and regulatory expertise, a customer-centric approach, and a commitment to continuous professional development. With cutting-edge technology and platforms, a focus on quality and regulatory compliance, Somru provides a one-stop solution for complex studies, partnering with clients to create a healthier tomorrow. Our innovative platforms include MABI, Agris, IntelliBeat, and Implato, which offers solutions for recombinant technology, recombinant antibody production, laboratory informatics, biosimilar develop, and ultrasensitive immuno-PCR. These platforms support a wide range of applications, providing our clients with reliable and efficient tools for bioanalysis and biomarker studies. Our bioanalytical services extend from discovery and preclinical work to clinical development and post-marketing studies. With expertise across multiple therapeutic areas, we support PK, ADA, NAB, biomarker strategies, ensuring compliance with global regulatory standards and contributing to successful drug development and approvals. Our technology platforms are designed to support diverse bioanalytical needs, including ELISA, MSD, flow cytometry, cell-based assay, and our ultrasensitive Implato platform. These tools are essential for biomarker detection, cytokine release, risk assessment, and cellular immune response evaluation, ensuring highest level of precision in drug development and testing. Somru Bioscience is ISO certified and holds multiple licenses, including PHAC and CFIA. We've currently undergone regulatory inspections, including GLP audit by Health Canada, and we have successfully supported our clients in submitting data to global regulatory authorities such as the FDA, Health Canada, and EMA. So with that summary of Somru Bioscience and some of our capabilities in the bioanalytical field, I will start my introduction for Dr. Kamala. So it is my pleasure to introduce Dr. Kamala, who currently leads the Clinical Bioanalytics Group at Dr. Reddy's Laboratories. Dr. Kamala is a recognized expert in developing advanced methods for pharmacokinetics, pharmacodynamics, and immunogenicity of therapeutic proteins, with a strong focus on supporting clinical studies. With a PhD in molecular and cellular physiologically and numerous awards, including the prestigious Angie Reddy Excellence Award, she has made remarkable contributions in the field. I've had the privilege of working closely with Dr. Kamala over the past 3 years in collaboration between Somru Bioscience and Dr. Reddy Laboratories. Together, we have advanced key bioanalytic projects, and her leadership and expertise have been instrumental in driving innovation and impactful research. Dr. Kamala is not only a brilliant scientist, but a dedicated mentor and strong advocate for diversity, equality, and inclusion, which makes her a valuable leader in our field. So with that, I will ask Dr. Kamala to initiate her presentation.
Thank you, Jessie, for the kind introduction. And first of all, I would like to thank Rafiq and Somru team for providing me this opportunity this afternoon today to share our work with you. So good afternoon, good evening, everybody who has joined to the virtual audience. And I'm Kamala, and I will start with my presentation now. So the title of my talk today is Mitigating Target Interference Challenges in Immunogenicity Assay to Detect Anti-Tocilizumab Antibodies. I know this looks like a very complicated title, but the aim at the end of my talk is we will understand what is target interference, what are immunogenicity assays, what is tocilizumab, what are anti-tocilizumab, and why we should be mitigating this target interference. So with that, can you go to the next slide? This is my disclaimer slide. So I think we can go to the next slide. The contents of the presentation today would be about the immunity and immune response. I will then introduce what are autoimmune diseases, and in particular, we will talk about rheumatoid arthritis. Then role of interleukin-6 in rheumatoid arthritis. As I said, what is tocilizumab? What does it do? What are anti-drug antibodies? How and why one should measure anti-drug antibodies using MST as a platform. And then I'll take you through a case study where we encountered challenges in evaluation of anti-drug antibodies, where there was a potential of getting false positives. And then we will close with conclusions and take-home message. Next slide. Okay, so before I get into the whole complex world of immunogenicity, I thought that it's always good to recap and go back to basics. So this will remind a lot of you about our Immunology 101. As you know, human body is very complex, and we have a very, very well-established immune system. And immunity is ability of an individual to be able to ward off or fight any infection. So broadly, immunity is divided into two types, innate and adaptive. I think, Jesse, you'll have to keep on clicking, yeah. So to differentiate, innate is, as the name is self-explanatory, it is what is within us, basically what we have it by birth. And it is present before onset of infection. And mostly these are like the natural barriers like our skin, mucous membrane, low pH in the stomach. So these are all innate. And this immunity is more like a short-term immunity, where this is the first response the body will have should it encounter any antigen or any germs like bacteria, virus. Now, adaptive is more of a long-term response that the body generates, and it's usually acquired after infection. I think a lot has been talked about and discussed, and then we all have lived through COVID era, so we know the importance of vaccination, and I'm sure most of us have taken the vaccine against COVID, right? And that is the kind of immunity that you get, which is adaptive, which is after getting the infection. Now, further, the adaptive immunity is divided into active and natural and artificial, active and passive. I'm not going to talk about all of that, but we will focus on the active, natural, adaptive immunity today. And this is something which will form the core of the talk. So anytime there is an infection like a virus, bacteria, or any antigen that is trying to enter our body or infect us, the first response is a cell-mediated response. And as you can see, there is a antigen-presenting cell. So this antigen-presenting cell takes the bacteria, virus, or antigen, and it presents to the T helper cell. And as you can see, this T helper cell will now activate the naive T cells. And as we go through the process, this will further activate different subsets of T cells like T memory cell, suppressor cell, cytotoxic cell, and helper cells. And all these small black dots are nothing but the interleukins or cytokines, which will further activate a second pathway, which will help in the elimination of antigen and lead to the humoral immune response, which is nothing but the B cell activation. And the B cell activation further can lead to memory B cells. These are the ones that come in handy with our vaccination. And the plasma B cells are the ones which will produce antibodies, right? So we've already, from the talk, we've understood how the antibodies are formed. And these antibodies are nothing but IgG, IgA, ME, or T. So the paradigm that we need to remember for the purpose of this talk is any antigen that comes, the antigen presenting cell will take it, give it to the T cells. T cells will try to eliminate it. While doing so, they will activate B cells, and B cells will form antibodies and memory B cells. So this is typically how the active adaptive natural immunity works. So this is very nice. So this is, as you can see, the human body is very evolved. It's a very complex, very, very well, I would say, streamlined process that we have within our bodies to ward off the antigens, basically anything that is foreign to us. Now I'll go to the next slide, which is actually on the other side of the spectrum. That is when our immune system breaks the tolerance, it loses the tolerance, and it is not able to distinguish between self and foreign. And it actually starts recognizing self proteins as foreign and mounts an exaggerated response. And that situation is known as autoimmune diseases. And one of the examples, well-known, well-studied example is rheumatoid arthritis, which I think most of you are familiar with. And as you can see in this diagram, on the right-hand side, you can see that the joint is really swollen, right? And if you look at the left side, the synovial membrane is intact and the joint capsule doesn't look distorted. But on the right side, you can see the migration of so many immune cells. There are T cells, there are plasma cells, osteoclasts, all sorts of cells, which actually are destroying the bone and leading to the bone loss and causing a very debilitating disease like rheumatoid arthritis. I'm not sure if you're familiar, but if you've seen somebody with rheumatoid arthritis, and I'm not exaggerating, like you can see my hand, they cannot even fold their fingers like this. Like even to do this is really, really painful and very difficult for them. So it's a very debilitating disease. And all because what happens, there are many postulated mechanisms, but the hallmark that of this disease, what people have understood is that our body generates citrullinated proteins, and this is a very normal process that happens. And some of the proteins that get citrullinated are like fibrin, vimentin, fibronectin, collagen. Now, this is a natural process, but for some of the individuals, their immune system recognizes these citrullinated proteins as foreign, and it generates anti-citrullinated protein antibodies known as ACPAs. So if you see these, if you take the serum of these patients and test, most of the patients who have rheumatoid arthritis have this ACPAs circulating. Again, as I said, this is the other side of the spectrum where there is an exaggerated immune response leading to an autoimmune disease. And as you can see on the right side, there are so many interleukins, interleukin 2, 15, 17. And as you can see, either it's leading to inflammation, bone erosion, or formation of autoantibodies, right? So all of this is manifesting into the joint or the bone destruction. Now, if we go to the next slide, apart from all these interleukins, there is another interleukin, interleukin 6, which is known to play a very key role in rheumatoid arthritis. And how it does it, the interleukin 6 binds to the interleukin 6 receptor. And then this receptor, which is on osteocytes, osteoblasts, synovial fibroblasts, T cells, they all activate RANKL, which is receptor activator of nuclear factor kappa B. And in the end, the osteoclasts are activated. So all we need to know is when IL-6 is overproduced and it binds to its receptor, it is causing the bone destruction, right? So let's go back next to the next slide. This is when in 2003, Chugai, a subsidiary of Roche, they actually developed a recombinant humanized monoclonal antibody known as tocilizumab, which binds to IL-6 receptor. So IL-6 needs IL-6 receptor to exert its signaling pathways, right? It cannot do anything on its own. It needs its receptor. So the first step is IL-6 has to bind to its receptor. And this receptor can be membrane bound or soluble. So when IL-6 binds to its membrane bound IL-6 receptor, it gets docked between GP130, and that's when the signaling happens. IL-6 can also bind to soluble IL-6 receptor, which is just, in the circulation. And this complex can travel and dock to GP130 on some other cell, and that is called trans-signaling, again leading to JAK-STAT pathway and cytokine storm. I think cytokine storm, tocilizumab, IL-6 were very, very common terms that we would have heard during COVID era, right? And in fact, tocilizumab was also prescribed for the COVID-19 patients. So what Chugai did is they actually developed this anti-IL-6 receptor antibody. And as you can see here, the tocilizumab binds to anti-IL-6 receptor, right? So if tocilizumab is bound to this receptor, IL-6 cannot bind. And if it cannot bind, it cannot exert the JAK-STAT signaling. Hence, there is blockade of cytokine storm, leading to the reduction in the inflammation and giving a remission to the rheumatoid arthritis patients. So tocilizumab is prescribed for patients who have rheumatoid arthritis, and this is the mechanism of action. So this is very nice. We saw that when the immune system is in overdrive, you have a lot of immune cells getting trafficked to the joints. So you give tocilizumab. Tocilizumab will bind to the IL-6 receptor, thus reducing the symptoms of rheumatoid arthritis. While this is one expected event, we'll go to the next slide and see what else will happen. In my first slide, if you remember, I showed you that when a virus or a germ or any foreign body enters, then the antigen presenting cells are recognizing it as foreign and they're presenting it to T cell. Same will happen with tocilizumab. It is not something that our body is producing. And it's a large monoclonal antibody. It's a 250 kDa monoclonal antibody. So when this is given either intravenously or subcutaneously, of course, our immune system will recognize this as foreign. It will present it to the T cell. T cell will in turn activate B cells. And that is when your anti-drug antibodies will be produced, right? Because the drug is tocilizumab and the body says this is foreign. So the B cell will produce anti-drug antibodies. Please keep in mind, for this process to occur, it takes minimally from anywhere between 7 to 15 days. The first set of anti-drug antibody class which is made is IgM followed by IgGs, right? These anti-drug antibodies can be further classified as binding and neutralizing antibodies. And so if you're familiar with the monoclonal antibody structure, this is the Fab portion, the Y, and this portion is the Fc portion. So the antibodies which bind, anti-drug antibodies which bind to the Fc portion, they are called binding antibodies. And the anti-drug antibodies which bind to the Fab portion are known as the neutralizing antibody. So both of them are not desirable because you can see already this tocilizumab is 250 kilodalton, to which if a large anti-drug antibody is bound, these are big complexes, right? So they will be cleared away from the circulation. You are giving tocilizumab so that the rheumatoid arthritis disease can be reduced. But when the body is taking this in, it's forming anti-drug antibodies and it's actually removing the drug from circulation. So obviously the patient will not benefit, means the efficacy of the drug will be reduced. Similarly, if this anti-drug antibody is neutralizing and it binds to the CDR region, the region or the epitope where actually tocilizumab will bind to the IL-6 receptor, again its effect is gone because instead of binding to IL-6R receptor, the anti-tocilizumab antibody has bound there and it's neutralizing its efficacy. So both of these are undesirable. In our world at Dr. Reddy's, we develop biosimilars to these molecules, and the program where we have the method that we have developed is for Dr. Reddy's tocilizumab, which is a biosimilar. So in our clinical trials, what we are evaluating is that the incidence of immunogenicity, whether you give Dr. Reddy's tocilizumab or innovator, it has to be similar, meaning Dr. Reddy's tocilizumab cannot induce more immunogenicity than the originator product. With this, we'll go to the next slide. So now that we know that its anti-drug antibodies are bound to be generated when you give a large molecule like tocilizumab, then the obvious question is, how do we detect these anti-drug antibodies, right? So if you visualize, tocilizumab can be given as intravenous or a subcutaneous injection. So once tocilizumab is given, as I said, it will take anywhere between 7 to 15 days for these anti-drug antibodies to emerge. So one of the techniques which is routinely employed in our field is a sandwich format, which you can see. The blue structures are nothing but the biotinylated drug, which is tocilizumab here, and the sulfo-tagged drug, which is against tocilizumab. So there is tocilizumab biotin, tocilizumab sulfo, and in between we will capture the anti-drug antibody because it's very specific. And biotin will bind to the streptavidin-coated MSD plate, and this will be a very specific detection. And this is a very routinely employed technique. But this is where the twist comes. And I'll go to the next slide. And this is where I'll take you to the case study. As I told you, tocilizumab was launched in market in 2010. That is like 14 years ago by the originator Roche. And as you can see on the slide, there are two graphs. Jesse, can I request you to use a pointer and take to the red lines and the blue lines? Yeah. So as you can see on the x-axis, there is time, and on the y-axis, there is serum IL-6 receptor concentration. So when a single dose of tocilizumab was given in normal healthy volunteers, what Roche found out was around day 15. Again, as I said, the anti-drug antibodies are generated around day 15. You can see that the IL-6R levels are highest. They are on around 250 nanogram per ml. And in two independent studies, what they saw by Zhang and Marcos et al., that these levels were anywhere between 250 to 330 nanogram per ml. Now, the normal circulating levels, if we go to 0 time point, you can see they are between 25 to 75 nanogram per ml. Meaning when you are not given tocilizumab, a pre-dose sample will have 25 to 75, but around day 15, which is a very common ADA sampling time point, there is a very high increase in serum IL-6R receptor levels. So the question is, okay, if the levels increase, how does it matter? We will see it in the next slide. What will happen is, if you see on the left side, I just showed you that we are sandwiching the anti-drug antibody between tocilizumab, correct? This will lead to the detection of true positive. But at day 15, which is a very common sampling point for ADA assays, when there is such high levels of IL-6R receptor, there is nothing stopping for this IL-6R receptor to go and sit between this, because it is what the tocilizumab is designed to bind to, and that would lead us to get false positive. And that is something that we don't want to detect, right? To the patient, we want to give the right information. So when we saw this literature, when we read this literature, it was very interesting. We did a lot of literature mining to see if Roche or any other groups, because I think we easily referred around 10 groups, a lot of publications are there, and we try to find out if anybody has tried to see the interference of IL-6 receptor in their assays. And interestingly, none of this was published. So we said, okay, then we will try and test our hypothesis that will increase in IL-6 receptor as high between 250 to 330 nanogram per ml. Will it impact our method or not? Because that was the hypothesis we wanted to check. So in the next slide, I'll just, for the audience who are not familiar with how we calculate our plate-specific cut points or confirmatory cut points, nothing to worry about. I will only request you to follow the lines. For this presentation, any bar which is above the line is ADA positive. Okay, so anything which is below the line is ADA negative. And in the first panel on the left side, you can see we tested 2 individuals, blue and orange, and in the negative control pool, we spiked the samples with varying concentrations of IL-6 receptor from 50 to 300 nanogram per ml. And both in our screening and confirmatory assays, we found that actually IL-6R level receptors were interfering in our assay. Meaning when there is no tocilizumab given, when there are no anti-tocilizumab antibodies, you can see on the right side, in the confirmatory assay, the purple graph is above the cut point. So that gave us an indication that our method was not target tolerant. And if in the patient or in the subject serum samples, if there are higher levels of IL-6 receptor, as already published, then our assay will actually detect false positives. And that was something that, of course, we did not want to have. So we started trying different strategies to mitigate this interference, because now we knew that there is interference in our assay. So in the next slide, the first experiment that we did was we said, okay, we have IL-6R receptor, we will spike, and we will spike varying concentrations of anti-IL-6R receptor. So we hypothesized that these both will form complex, and after sample processing, we will remove the IL-6R in our samples. And we tried varying concentrations of anti-IL-6R antibody from 10 to 4,000 nanogram per ml. And again, as I said, anything above the line is positive. We made samples having varying levels of IL-6 receptor, 100, 750, and 500. As you can see, the orange bar, where there is no IL-6R receptor, except that all the samples were above the cut point. So instead of reducing the interference, we actually started getting very high signal. And we saw that using this blocking antibody, we would not be able to remove the interference. Then we moved on to our next experiment. In this experiment, we followed the paper by Chen et al., which was published around 2019, if I'm not wrong, where they also were experiencing interference in their assay from the target. So what they had done, or what in their publication was, they used anti-target antibody, and they tried low basic pH, and they varied the conditions. So we tried to simulate the same. And as you can see, except the negative control, where we have not added anything, when we took samples, the negative control with IL-6 receptor and anti-IL-6R antibody, we again were getting signal positive. That is, all the bars were above the cut point, and this strategy also was not working out for us. Then we said we did not give up. We tried the next experiment. As you know, most of you are familiar that in our immunogenicity assays, we mostly do acid dissociation. The reason that is done is because the drug and the anti-drug antibody are always bound very tightly, right? So we use acids like 300 millimolar acetic acid to dissociate. So we hypothesized that because we are using acid dissociation and we are making these complexes free, that's why maybe there is more and more of IL-6 receptor which is free, and maybe that is interfering in our assay. So we created samples, which is our positive controls, PCH, PCL, negative control, and samples with, again, spiked with IL-6 receptor 50, 100, 350. And whether we did acid dissociation or we did not, in the end, there was still interference, meaning all the bars were above the cut point. So none of these strategies worked. And only one key takeaway from this slide is, at this point, the MRD of the assay was 1/20. MRD is nothing but minimum required dilution, where we dilute out the interference from the matrix. So then the only thing that was left to try out for us was, what if we increase the MRD? Will it help us reduce the interference? So in the next slide, you can see that we tried different MRDs, 20, 40, and 80. Along with this, we also used a very high saturating concentration of IL-6. If you remember in my previous slides, I told you IL-6 binds to its IL-6 receptor, right? So we said we'll increase the MRD and we will add very high concentrations of IL-6 to see if it quenches the IL-6 receptor. And this is where our eureka moment was. And as you can see, as the MRD was increasing, if I can show you these samples, particularly in blue, pink, and orange, where the negative control pools are spiked with the 350, 750, and 100 nanogram per ml of IL-6 receptor. As the MRD was increasing, you can see the bars are below the cut point. So it was finally at 1:80 MRD with 5 microgram per ml of IL-6. We were actually were able to achieve the target tolerance for the method. Now this experiment was done in pool samples. So we wanted to test this hypothesis and see, will this result be reproducible if we try it in individuals? So if we go to the next slide. We took 5 individuals and then we spiked them with 500 nanogram per ml, 350 and 250 nanogram per ml of IL-6 receptor. Again, we ran the samples at 1:80 MRD in presence of IL-6. And as you can see, we got target tolerance for 5 out of 5 individuals at 350 nanogram per ml and 4 out of 5 at 500 nanogram per ml. So finally, we were able to remove the target interference by increasing the MRD and adding more IL-6 in the samples, in the master mix. So in the next slide, once we knew that the method was working, we tested the other parameters which are routine for immunogenicity assay. And the other important element was to establish the stability as the specificity of the method. As I said, one, we were adding IL-6 in the master mix, so we wanted to make sure that there's no interference and the method is still detecting only anti-tocilizumab antibody. But what is also published by originator is when tocilizumab is given, the IL-6 levels go up. Also, not very high as IL-6 receptor, but from 10 picogram to 30 picogram. And in this experiment, you can see we tried again 5 different individuals with 500 nanogram per ml of IL-6. And again, all the bars are below the cut point, only the positive controls are above, which confirmed the method is very specific. And of course, the next experiment was to test the drug tolerance. And as I said, at Dr. Reddy's, we are a biosimilar company. So it's important that our method performance is similar between Dr. Reddy's tocilizumab, Actemra, which is a Europe product, and Actemra, which is the US reference product. And we were able to achieve 100 microgram per ml of drug tolerance with as low as 50 nanogram per ml of positive control. And in the next slide, the most important experiment for us was to establish antigenic equivalence. What it means is we need to show to regulators that our method, whether it is detecting anti-drug antibodies because of the innovator product or Dr. Reddy's, the method performance remains the same. And that is what is shown in this graph. We tested the high positive control, medium, low, and dissociation positive control made with all the 3 drug products. And these 3 drug products were used in the confirmatory assay. And with the overlapping lines, what it clearly says is that the assay was similar. The performance was similar. It was not biased because of any drug product that we were testing in this method. We also tested sensitivity of the assay, the positive controls, when they in the confirmatory assay, when they were inhibited either by Actemra, Actemra, or Dr. Reddy's tocilizumab, all the curves are overlapping. The sensitivity was between 24 to 12. So the method is similar. There is same equivalence. And lastly, even the drug tolerance between the 3 drugs, the Actemra, Dr. Reddy's tocilizumab, and Actemra, as you can see, All the 3 drug products were able to tolerate 1000 microgram per ml of drug at 100 nanogram per ml of positive control, which established that this method was not only target tolerant, it was sensitive, it was drug tolerant, it was specific, precise, and of course, the antigenic equivalence was also proven. And with that, the final format of the method is that, as you can see, in the sample, when a subject is given single dose of tocilizumab, you can expect tocilizumab, anti-tocilizumab, and IL-6 receptor. When we do acid dissociation with acetic acid, you can see all of those are dissociated, they're separated. And then we neutralize them in presence of high concentrations of the serum IL-6. And the IL-6 is quenching the IL-6 receptor away. And then in the end, what we are detecting using MSD is only anti-tocilizumab antibodies. And the next slide. So this brings me to the end of my presentation. So the take-home message is that human body has very well developed immune response, and they can be broadly classified as innate or adaptive. But sometimes body does recognize self as foreign and mounts an autoimmune response. Upregulation and production of cytokines like IL-6 is directly correlated in rheumatoid arthritis. And monoclonal antibody tocilizumab is known to be effective in rheumatoid arthritis. However, administration of monoclonal antibodies can also lead to unwanted immunogenicity. Hence, the regulatory agencies mandate use of validated immunogenicity methods to assess the incidence of immunogenicity during a clinical study. So it's really important for us as bioanalytical scientists that we develop precise, specific, target-tolerant, drug-tolerant selective assays, which should not give false positive. I think I tell my team that it is our moral obligation. We owe that subject or patients each microliter that they trust in us and which are collected as part of clinical trial samples. I think it's very, very important that all of us make sure that our assays are not producing false positives. And I'm happy to share this work was published this year in July in Bioanalysis. And with this method, we were able to complete successfully 2 of our phase 1 studies with SC and IV presentations for Dr. Reddy's tocilizumab biosimilar. And with that, to the last slide of acknowledgements, I would like to sincerely thank my co-authors, Mamta, Kaila, Hema, Sagarika, Soumyaranjan, and Vikas. We were the group who were part of the authors for the publication. And on the right, you can see my young, dynamic, energetic scientists, some of who have joined today's presentation. They keep me on my toes. They make sure that our scientific curiosity is always up. And of course, I would like to thank my boss, my mentor, Dr. Narendra Maharaj, who's always supporting us and helping us in all our scientific endeavors and pursuits. So with that, I come to end of my talk, and thank you so much for joining in and listening to me, and I'll be happy to take questions now.
So at this point, we want to open the floor for some questions. I do have a couple questions that were initially received it here in the chat. So one question being, when trying the blocking anti-IL-6R blocking antibody, did you test various blocking antibodies, or was that a singular attempt?
Yeah, we actually tried from 2 vendors, Merck and R&D, and the results were similar.
Okay, great. Another question that I do have regarding the blocking antibody is, what would cause the signal to go up when you saw that increase with the anti-ILR6?
Yeah, I mean, initially we thought it was maybe a method artifact, but I think, as I told you, and this is a caveat, and one has to be really careful with using these anti-IL-6 or any anti-blocking antibodies, because you can imagine anti-IL-6R is nothing but tocilizumab. Anti-IL-6R is nothing but the anti-tocilizumab antibody, right? That's what we're trying to detect. So we hypothesize that maybe the epitopes or the CDR regions were same. So where the anti-drug antibody should bind is where the blocking antibody was binding, and instead of quenching the IL-6 receptor, it was actually forming the sandwich. So it did not serve our purpose, and this is also is well documented that that it's really important that you thoroughly characterize your reagents. I mean, what I'm trying to say is, even if this blocking antibody would have worked for us, we would have definitely done experiments to make sure that that it was really removing IL-6 receptor from the samples, and it was not leading to any false positive like how we saw. But that's very common finding. Yeah.
Okay, great. In regards to the basic pH, I know that the acid treatment is quite common. Can you just explain the thought process behind attempting the blocking with the basic pH?
Yeah. So as I mentioned, we referred Chen et al. I believe it was 2019, Chen et al. Where the group had similar issue for, I believe, for a IgG4, where they were seeing interference with their target, and this target was leading to false positives. So what they did is they used the combination of the blocking antibody, which again they titrated, and for them it worked out. It did not give a false positive signal like us. With in this condition, and more of it is detailed in the manuscript, is we tried various combinations of 16 millimolar of tris versus 1 millimolar of tris. And then we played around with the sequence. When do you add the anti-IL-6R antibody, and how do you change the pH, in hope that maybe we would be able to get rid of the interference like Chen et al. Did. It worked out for them and not for us. But the more of the details were basis that manuscript that we had referred at that point. And of course, we had tweaked it for our experimental design, but the thought process was that maybe it will break the bonds, and we will be able to remove the IL-6 receptor away, so that the sandwich is still bound by the. That was a basic premise, but of course that did not work.
Okay, great. You also mentioned doing some acid treatment. During that acid treatment, were you using any solid phase to remove any of the IL-6R?
No, we did not use any solid phase treatment. And also one thing that I wish to add is, when I say MRD of 80, when this work was done, that was way back in 2020, where the thinking was, at least in our group, we thought that the MRD we would report is only the first dilution with the acid, but there are subsequent dilution steps. So in the end, the MRD is around 243 something. So we did not use any solid phase. It was just, as I showed you in the picture, this was all in solution.
Okay, great. That actually answers another question that had come up, was the sensitivity reported with the initial MRD before or after all of the subsequent dilutions?
So it is in our validation report, we have actually done both. I mean, okay. We've done it with 243, which is the final dilution, and we've said that at need, this is what you would have. But since we've used a monoclonal PC, it was really, really sensitive. Even at 1:80 or in the end 243, we still had met the sensitivity requirement. Yeah.
Great. So speaking of sensitivity, often we find that when we perhaps fix one problem, we have created another. Did you have to sacrifice some sensitivity to kind of resolve this? And if so, what would how much and how did you kind of resolve this sensitivity?
Actually, we didn't have to. As I said, the monoclonal PC that we used was from Biorad, and it was really very, very high affinity. I mean, I think at 1:20, maybe it was like 2 nanogram per ml, which sometimes worries me, right? Because I'm not even sure if that's real. So what we were seeing at 1:80 was a little bit more realistic. What I've not laid down here is, but the subsequent antigenic equivalence experiments that I've shown you, they are with polyclonal antibody, which was generated against Dr. Reddy's tocilizumab. And there also the sensitivity of the method was retained. And that was like our, I would say, that was our innovative point, that despite increasing the MRD, we did not compromise on sensitivity, which was expected. We still were very sensitive assay. And that sensitivity was comparable between the monoclonal and polyclonal PCs.
Great. So those were some of our written questions. I just want to open the floor again. If anybody wants to ask Dr. Kamala any additional questions.
Hello, Dr. Kamala. This is Kazi. Thanks for the wonderful presentation and explaining some of the basics that I'm just going through right now. So it was really helpful. I just had one question about your presentations, or especially like towards the last few slides where you're showing comparison between different drugs. To be just more precise, like I didn't see any stats, like, or any error bars on your bar. So I was just wondering, like, were those experiments replicated, or were those just a single run?
So, Kazi, thank you for that question. And as I said, the bar graphs were more for the visual representation. Typically, since immunogenicity assays are quasi-quantitative, it's just a yes or no, right? Is it above the cut point, at cut point, or below cut point? So there are only just numbers that we are trying to translate in terms of either percent inhibition, just for visual representation. So most often, except cut point and sensitivity, like all the other experiments are done once, barring precision. So like the drug tolerance experiment is just done once, and the representation can be signal to noise ratio or percent inhibition or just the raw value. So that's why no error bars are indicated there.
Okay, thank you so much.
You're welcome.
I do have one question. Thank you for the awesome presentation. Did you test that method in rheumatoid factor matrix?
Yes, we have tested this in RA matrix, and the similar performance was seen.
For similar performance, awesome. Thank you.
Yes. I think, Jesse, you're speaking on mute.
Sorry, I was just asking one last time if anybody else has any additional questions they would like to ask Dr. Kamala. I guess in that case, thank you so much, Dr. Kamala. That was a very wonderful presentation, very informative, from the basics of our Immunology 101 to the intricacies of the development and troubleshooting that you were able to get through. So thank you very much.
Thank you, Jesse, and thank you, everybody, for your wonderful participation.
Great. I guess that will conclude our webinar. I think we are right on time here, just under our hour point. So thank you, everyone.